In this video, we will discuss about a technology that is FRET. Uh, it is abbreviated as Fluorescence Resonance Energy Transfer, and so we will understand what is the principle behind this FRET and one of its application that is used to detect the Ca2 plus level within the cells. So let us quickly go through how this FRET technology works. So consider a protein and that has a this we can say reporter, it is CFP reporter. So and the we can say energy at which it is excited and emitted is this 430 nanometer and 475 nanometer respectively. So whenever the light of 430 nanometer is uh, is thrown to this CFP reporter, it will get excited and emit the fluorescence at 475 nanometer. And however, this protein B has this YFP, and this condition of this YFP is that it will get excited by this 475 nanometer fluorescence, and then it will emit the fluorescence at 525 nanometer. So when these proteins are separated apart, then this emitted wave then this emitted you can say this CFP emitted wavelength will not be able to excite this YFP. So, and however, when the proteins A and B interact to each other, or when they are close to each other, what happens is that this CFP emission, there is energy transfer occurs. In this case, what happens is that this CFP emission, uh, this fluorescence that is emitted from CFP, that is this 475 nanometer, will be used to excite this YFP. So, and we will get the signals from the YFP, that is at 525 nanometer. So how we can differentiate these two conditions is that in these conditions we will only see the signals of this 475 nanometer. However, in this case we will see the reduced signals of 475 nanometer compared to the first case or the control case, and we will also see the additional signals of this 525 nanometer. So in this case we can distinguish whether these two proteins are separated to each other, or these two proteins are close to each other. So this is known as fluorescence resonance energy transfer. That is, energy is transferred from the CFP to emitted emitted light of the CFP is used to excite this YFP reporter. So this is the basic idea or principle behind FRET, and how this FRET can be used uh, for for some biological application is explained by this figure. And in this case, what the problem, let us first understand what problems this FRET is addressing. So the calcium levels, this FRET is able to detect the calcium levels within the cells or within the organelles. So uh, before this FRET, it is difficult to identify the calcium levels in the cells or in the organelles because of many reasons. We will not go into those reasons. We will understand how this FRET helps in identifying the Ca2 levels within the Ca2 levels within the not yeah Ca2 levels within the cells. So this is the reporter that they have used. In this case, this uh, they have used this CAM and M13, and that helps in connecting two GFP reporters. And so what happens in the normal case is that only the light of 370 nanometer, fluorescence of 370 nanometer is used. So in this case we will see the signals of around uh, this 480 nanometers and 480 nanometers when these two reporters are separated apart. However, this linker is designed in a way that whenever the Ca2 plus is free Ca2 plus is available within the cells or in the organelles, it will bind to this linker that helps in the conformational change in a way that these two reporters are close, distance between these two reporters are reduced, that we can see here, and the energy transfer, fluorescence resonance energy transfer can occur. So in this case what happens is that when we when we provide this excited fluorescence wavelength of 370 or 440 nanometer, so instead of emitting this 480 nanometer, that 480 nanometer will be used to excite this YFP, and this we will get the final signals of this 510 or 535 nanometers. So in this case we will be able to distinguish whether the free Ca2 are present within the cells or within other separate organelles or not, by just observing the signals, final signals, whether we get this 535 nanometer signals or the 480 nanometer signals. In this case also we will get some 440 nanometer signals. That could be because of some of the reporters might not be able to find free Ca2 plus level, or this complete energy transfer might not have occurred. So however, we will be able to distinguish these two cases, that is the one in which Ca2 is bind with this CAM, and the one in which there is no Ca2 binding occurs, with the help of just measuring these fluorescence signals. So this is how the FRET technology, how the FRET principles works, this is how the principle of FRET works, and how this FRET can be used to solve one of the biological problems. So that's all for this video. If you need to understand more about this FRET technology and how it works, you can go through the references that has been provided in the description. Thank you for your time.